Alison Bentley. She comes from NIAB, UK, and she's the program leader for trade genetics. Actually, I'm impressed with Alison. She was in Punjab, and she got stuck in Haryana for 2 days on the road, and she made it to here. Actually, I was thinking maybe she's still there. Anyways, I mean, adventurous trip for her, first time in India, and the real picture of India. So anyway, Alison got her PhD from University of Sydney, Australia. Then she moved to NIAB in 2007, and she's right now trade genetics program leader. And in addition to that, she's handling a lot of other activities as well. She's involved in a program called Synthetic Wheat Program. She's also chair for UK Monogram Small Grain and Grass Community, chair for the Wheat Initiative for Expert Working Group on Breeding Methods and Strategies. And she's also a team leader for Agriculture and Food Security team of University of Cambridge doctoral training program. I think she is going to talk about the pre-breeding and genomics, how to link those two in wheat. And yeah, she's here till tomorrow afternoon. And if someone wants to talk to you one-to-one, just let me know. Alison, thank you.
Thank you very much for the introduction, and I too am very surprised to have made it to Hyderabad. I thought I would be living the next week or so in Haryana, but I'm very happy to be here and to be presenting on our wheat pre-breeding program. So I know wheat's not a big crop in this region, but hopefully today I wanted to give a snapshot of the program that we've been running for almost 10 years now. On wheat pre-breeding, which was really a new thing in the UK when we started out in 2007, and we've invested very heavily in the creation of new germplasm, the application of genomics for selection, and in various other areas that I'll talk about today. So NIAB, for those of you who don't know, is an independent plant sciences company, so we're completely publicly funded. And we live a very hand-to-mouth existence based on research funding. We're based in Cambridge, so we have a large field site on very lucrative real estate outside the city centre of Cambridge. So this is our field site, and we run huge numbers of trials. So we're really focused on applied wheat research. And we have very close connections with the breeding companies as well as in academia. So in my talk today, I thought I would give a snapshot of the program, and this includes our work on characterizing adaptive response in wheat, which is a very simple experiment on paper, but has turned into something much more complex. Our work on resynthesis and on the use of highly recombined populations called magic populations, and within our program, we have a very strong emphasis on quantitative genetics, so understanding the quantitative genetics behind complex traits, and I'll talk about this at the end of my presentation. So starting with our work on flowering time, this is work I started when I joined NIAB in 2007, and it was a very simple question we were asking. And why we're interested in adaptation is because why we're interested in flowering time is that it is the central point of adaptation to the environment. So wheat is grown over a wide geographic range, and it's flowering time which adapts wheat to the specific environment in which it's grown. So this is a picture of Europe, and you can see in southern Europe we have early flowering wheat varieties, and this has been established for a long time. So if you grow early flowering or photoperiod insensitive wheat varieties in southern Europe, you get a yield advantage. And this is because you have a hot, dry finish to the season, so a later flowering variety will encounter drought and heat stress. Whereas if we look in the United Kingdom, where we are based, early flowering types actually confer a yield penalty. And this is the effect, so we have a very long and a very lush growing season, plenty of rainfall, not very hot conditions. So in those environments, early flowering types will flower too early and not take advantage of this long, lush and easy growing season. So actually in this region, so in the UK, all wheat varieties are late flowering photoperiod sensitive types, which is in contrast to southern Europe where you have photoperiod insensitive early flowering types. And this has been known for quite a long time. It's how early farmers selected and adapted wheat to the environment to which it spread. But there was very little known about the genetic control of this adaptation to the environment. And so a large part of this is controlled by the photoperiod 1 gene, PPD1, and the gene was cloned in barley back in 2005, and then in wheat as well. And the major contributor to photoperiod insensitivity in wheat is this PPD-D1 2 kb upstream deletion, and this causes this early flowering photoperiod insensitive phenotype, which you see this difference between Southern Europe and Northern Europe. And as well as that, if you survey the photoperiod 1 gene on the A, B, and D copies, remembering wheat's a hexaploid, so it has 3 copies of the PPD1 gene, we see there's quite a large amount of additional allelic variation for the photoperiod 1 gene. So what we wanted to do in this study was to put each of these allelic variants in a common UK-adapted background and assess under common conditions the effect of this photoperiod 1 gene. So this is a very simple experiment to create near-isogenic lines to allow us to understand the effect of allelic variation at this gene on flowering time and the trade-offs with yield and other agronomic effects. So we ran this program as a large marker-assisted backcross selection program. I had previously worked in haploid fungi, so when I came into the program and needed to develop diagnostic markers in wheat, I spent quite a lot of time amplifying the wrong copy from the wrong genome. So part of this was developing the marker system to allow us to run the program. And at the end of this, we had field nursery plots, which allowed us to assess the flowering time response. And then we've continued this into looking at the yield response. So a very simple question: can we characterize the allelic variation at the photoperiod 1 response gene? And so we did this in the UK quite successfully, and we were able to provide that material to our partners in the breeding industry to use in their programs. So I should mention that in the UK, all of the breeding occurs in the private sector, so we're not able to produce varieties, but all the information we generate is passed into the private sector and used by the breeders in their breeding programs. So we then have extended this study beyond the 5 years that were funded to create the material to further understand this difference between Northern Europe and Southern Europe, as was shown previously, but with this greater array of allelic variants at the photoperiod, at the PPD1 gene. I know Jana, from talking to her this morning, is very skeptical about the use of glasshouse phenomics facilities. In this case, we used the glasshouse phenomics capability at the National Plant Phenomics Centre in the UK at IBIS in Aberystwyth. And we used it because we wanted to know the precise developmental differences between the allelic variants for this one single gene that we had identified and put into a common genetic background. So in this experiment, we're saying we've got one common genetic background, a library of allelic variation. Can we use an automated system to understand on a developmental level what the differences are and how these interact with different combinations of heat and drought stress? And this work is still ongoing. And we had some really interesting results using our photoperiod 1 near-isogenic lines. And these were mostly around the PPD-A1 gene, which was only recently characterized from durum wheat, and which we were able to bring into our marker-assisted backcross program via synthetic wheats. So this is a novel photoperiod response variant on the A genome, which gives in the UK an earlier flowering phenotype than the classic PPD-D1 genotype. But whereas PPD-D1 will confer a yield disadvantage, which is what's been known for a long time, PPD-A1 doesn't confer the same yield disadvantage. So it has an earlier flowering phenotype and no yield penalty. And so this is really interesting to our breeding partners because you're able to bring the flowering time earlier but not compromise the yield. And now we need to understand what the mechanism for doing this is. And we also break some linkages. So the PPD-D1 gene is associated with is linked to a reduced height gene called RHT8. So if we have alternative sources of earliness which don't confer yield penalties in our target environments, we can use these quite successfully to bring in new sources of adaptive response. So we can also use the phenomics data to try and understand what's been happening developmentally across the time frame of the experiment. And this is the changes in the color that is picked up by the cameras in this system. So this red arrow represents the start of our stem elongation. So we see this plateau in the green part of this curve. And equally at flowering time, so when the plant's reallocating its resources, we pick up some changes in the color signal. And at the moment, we're trying to combine this with transcriptomics data to understand what's happening primarily in the senescence pathway, and if we can use this as a marker for understanding the transitions that are occurring in these variants of the adaptive response gene PPD1. So that program that I've described was a very simple question: what are the effects of these allelic variants in a common genetic background, and how can we use those to adapt wheat to its environment and understand trade-offs with yield and different growing conditions? In the next part of my talk, I will talk about our resynthesis program, which we have been running since 2007. So our first part of this program, I'll go here first. So wheat, for those of you who don't know, is a hexaploid. So it combines in its genome the genomes of 3 ancestral grass species. The first 2 genomes came together about 100,000 years ago. So these are the A genome from Triticum uratu and the B genome from a relative of Agelops speltoides. So this chance hybridization event occurred a relatively long time ago and gave rise to Triticum dicocoides, which is called wild Emma wheat. Wild Emma has been subsequently domesticated to a form called domesticated Emma, Triticum dicoccum, and then also to Triticum durum, so pasture wheat. So these tetraploids have been evolving over a period of about 100,000 years. A second chance hybridization in nature occurred to bring in the 3rd genome from the wild goat grass Agelops tauschii. And it's predicted that this event occurred only a few times. So this is the genetic bottleneck in wheat domestication. And this occurred about 10,000 years ago, so it hasn't had this long time frame of domestication. So over many years, CIMMYT worked on this process called resynthesis, where you recreate these hexaploid genomes. And when we started our program, we requested from CIMMYT their large collection of synthetic wheat. So they have over 1,000 of these synthetic or resynthesized wheats. From that, we selected 50 of the most genetically diverse, and we crossed those into UK-adapted parents, I-19 and Paragon, a facultative and a spring wheat. And we ran this very much as a phenotypic breeder selection. So we made back-cross populations as well as beginning selections from the F2 generation. And over subsequent cycles of selection, we were able to select a core set of 300 of these lines, which represent the best phenotypic traits coming through from the CIMMYT synthetic material. And these are type of characteristics that we're looking for, so an increase in our yield components. Here we have Xi19, which is one of our parents and the primary synthetic, and this is one of the derivatives from that population. So we're seeing bigger ears, larger numbers of yield components, and these are the types of things that we were looking for, bringing new diversity into UK breeding. And in that program, we were basing our starting material on material generated at CIMMYT over several years. And we had a few issues with doing this, and also the types are very spring-like. So in the UK, 95% of the wheat grown is winter wheat. So we asked the question, can we actually make more targeted synthetics for a European breeding context? And so this is what we set about doing within a large UK partnership called WISP, which is the Wheat Improvement Strategic Program. And this is a very large program of public sector pre-breeding, which is funded by our BBSRC, our bioscience funding agency. And it brings together our work on synthetics, work on landrace material from the John Innes Centre, and work on ancestral gene integration, so from a wider gene pool. And these are linked together through common phenotyping and common genotyping, as well as an interaction with breeding partners who are obviously producing varieties. So the part of the program that I run is to look at the D genome, so this goat grass genetic bottleneck, and to see if we can use resynthesis to bring in new diversity for breeding. So what we do in this case is we take an elite durum wheat as the tetraploid female. We pollinate it with pollen from goat grass or Agelops tauschii, which is a very weedy looking species. Very few occasions we are able to generate an F1, which is an unstable triploid. So we rescue the embryo from this, artificially double the chromosomes using colchicine, and then at the end of this cycle we have a synthetic wheat or a resynthesized version of the hexaploid. And in the CIMMET program they generated thousands of these, and so far we've generated 50. So this represents at least 5 years and 7 crossing cycles to generate 50 of these synthetic wheats. And we make all of our material completely publicly available and free of intellectual property, so we're able to distribute it as widely as we can. And one of the other reasons for making new synthetics is that there's a huge amount of geographic and genetic diversity which is present in these goat grass accessions. So we collected goat grasses from ex situ gene bank collections, and this figure shows you the geographic distribution of our Agelops tauschii goat grass collection. So bread wheat was domesticated here in this region to the south of the Caspian Sea, and these are represented in the yellow dots. And the previous work by CIMMET to create synthetic wheat used goat grass accessions from this same geographic region. But you can see that the range of these goat grass accessions extends quite a distance outwards across the Tibetan Plateau. So there's a huge amount of additional geographic variation that we wanted to capture. And when we genotype them, so this is based on some Cas markers we've developed, you see this very tight cluster characterizes the goat grass accessions in this region where wheat was domesticated, and all of the CIMMET synthetics and our elite bread wheat fall into this cluster, and this cluster here represents this additional geographic diversity. So what we're aiming to do in our program is to capture as much of this additional genetic and geographic diversity in an effort to increase the genetic variation that's available for wheat breeding in Europe. And in our previous program, we used purely phenotypic selection, and we're only now at the stage where we're going back to the material, genotyping it and saying, what have we actually selected? In this program, we have had and continue to have an assortment of genotyping tools available via our partnership with the University of Bristol, Keith Edwards' group there. So we have and use extensively Cas markers for characterizing all of the material that we create in the program. We use to a lesser extent a 90K SNP array, which was developed a few years ago by a consortium. And most recently, we've been involved in the development of a new Affymetrix array, which has 820,000 SNPs on it. So we exome captured quite a number of our tauschii accessions and used these in the creation of the discovery array and subsequently in the validation. So we have a lot of, whereas in contrast, previously we were relying on phenotypes, we're now relying quite heavily on being able to genetically track our material. And again, all of our genotype data is made publicly available via Serials DB, which is the UK database of genotypic and marker information. We also characterize our tauschii or goat grass collection for functional diversity, so not all diversity is good, some will be terrible, and we have specific traits that we're interested in for various reasons. So coming back to the photoperiod 1 story, if you sequence the photoperiod 1 gene from the Agelops tauschii, there's additional allelic variation that's not being characterized, and this also equates to variation in flowering time. So we can bring through some novel photoperiod response types, as well as some novel vernalization types through this D-genome donor. Here's an example for the number of tillers produced by a plant. So we're interested in this from a hybrid production aspect, as well as from a yield component point of view, and we have huge amounts of exploitable variation. And yellow rust is a huge disease problem in the UK, so we also screen the material in seedling screens with multiple races, as well as in field nurseries, which allow us to identify a small subset of the population which is resistant to yellow rust in the field. And so we can move these forward in more targeted combinations rather than just relying on geographic or genetic diversity. So the beginning of the story is really the creation of these resynthesized wheats. Following that, we do a systematic crossing to Robigus, which is a UK winter-adapted variety, and a UK spring variety. And within this program, we're generating 10,000 BC1F5 pre-breeding lines. So obviously this is a huge amount of unselected material. But at the start of this program, the breeders and the funders asked us to introduce new genetic diversity to wheat. So now we're saying, here's your 10,000 lines. They're as diverse as we could make them. And the question really then remains, what do you actually do with 10,000 unselected lines? One answer is that because they're crossed to two common parents, we can analyze them from a quantitative genetics standpoint if we can phenotype them. We can analyze them as a nested association mapping panel for any of our traits of interest. But more importantly, we go back to our original approach. So we put all our residual seed in the field and we make breeder selections on all of the early generations. And so these are really the core set of the diversity that we're capturing. And we make these bulks and selections fully available as well, because people are much more likely to characterize a small subset of material than to request 10,000 pre-breeding lines. And these are just some pictures from the field this year. So we have some interesting anthocyanin producing types. Some types in our early generation, so these are BC1F3 families that look very much like an elite adapted UK winter wheat. And this is an A4 page. I cut off the bottom, but you can see we have some very large ear types comparative to our standard varieties. And obviously there are many trade-offs associated, but this is the type of diversity that breeders can use and take forward into their programs. As well as developing our 50 synthetic wheats, we're developing chromosome segment substitution lines, which are essentially a library of introgression fragments from a single goat grass accession. So we selected our goat grass accession based on exome capture, which we did on a range of our accessions to be the most diverse accession. Again, we run marker-assisted backcrossing, and this is really aided by our ability to use highly saturated Cas markers. And this is an example of what we're doing in the intermediate generation. So we're selecting heterozygotes which cover the 1D chromosome. So we basically have a library of lines, common genetic background, each with small overlapping segments of this single donor, one of our goat grasses. And again, so this is the starting point for your trait dissection and the start of fine mapping for traits with genetic controllers on the D genome. So now I'll talk about our magic population, our work on magic populations. So resynthesis aims to bring in new diversity, and magic populations aim to reassemble diversity that's present in the founder varieties. So magic, for those of you who don't know, stands for multi-parent advanced generation intercross populations. So these start at the beginning with some number of founders. So we have two populations at NIAB, one created from 8 founders, as you can see here, and another created from 16 founders. And so the basic premise is you put 8 founders in at the top, shake them up in a bag, and then extract highly recombined rills at the end. So you cross these 8 founders in all possible 28 two-way combinations, all 210 four-way combinations, and then all 315 eight-way combinations to close the loop. So everything is intercrossed with everything else at each generation. You then derive F5, F6, or F7 rills from the end of this process. So this is a huge investment in the creation of a genetic resource, and just this pedigree scheme here represents about 6 or 7 years of intensive crossing, soul-destroying crossing in a glass house, and remembering that this is winter wheat, so it requires vernalization for 8 weeks. So this was a significant investment of resources to create the population. But we believe that it has been a very useful thing to create. So we were funded simply to make the population, but as with the creation of all germplasm resources, creating the germplasm is really the starting point from which to go on from. And in our first publication, so we started the work in 2007 and got a relatively low impact paper in 2014, just to indicate that we're not doing it for the glory. But we were able to map this gene, which is the presence or absence of orns, these structures on the ear. So we were able to map that within the population, extract a high quality diagnostic Cas marker in a single step. So it works for high precision mapping within the population. As I mentioned, yellow rust is a big disease problem in the UK, and so we're using this population to try and characterize the genetic controls of resistance. So within our population, our most resistant parent is a parent called Hereward. Our least resistant parent is a parent called Robigus. And when we look at the distribution of resistances in our progeny, we see material that's more susceptible. And we also see this very interesting transgressive segregant subset, which is highly resistant to yellow rust. So there's no yellow rust resistance in UK winter wheats, but here we have a subset of our real population which is highly resistant. So we're able to map that effect, so we map it to 3 loci, all with relatively good effects. And then if we look at the profiles in our founder varieties, you can see that none of our founders carry more than one of these resistant alleles. So Claire, for example, is a variety that had durable rust resistance. So I'm not a rust pathologist, but rust pathologists will tell you that durable resistance is a resistance that doesn't break down. So Claire had durable resistance and no longer does. So obviously it wasn't durable resistance, but it's interesting. So alchemy is identical by descent to Claire. And it's interesting that even though Claire's resistance is broken down, it's still contributing a relatively large effect resistance allele to these transgressive segregants. So this is more like a quantitative resistance. So even though that is not resistant, it's able to contribute a resistance allele. And so our transgressive segregants combine all 3 resistant alleles. So this is classic transgressive segregation due to this dispersion of the alleles in the parents that are brought together by these multiple rounds of recombination. So you wouldn't be able to extract this kind of recombinant from a classic biparental population. So the advantage of MAGIC is the recombination of these alleles into individuals, as well as our ability to map those, so we can map the effects and extract the individuals in a single step. So this is really the power of using these multi-founder highly recombined populations. We have additional diseases which we do this for. So this is Fusarium head blight, which is an emerging disease in Europe. In the UK, it's quite a big disease already in France and Norway and parts of Germany. So again, we have transgressive segregants within our population. I should say this is one of my long-suffering PhD students. So the disadvantage of MAGIC is you have 1,000 RILs. So you need to be able to phenotype, accurately phenotype at least 1,000 RILs. So if you're doing something like Fusarium head blight where you need quite a lot of replication, it is a fairly soul-destroying process. But for a very good phenotype and a very efficient mapping at the end of the process. I thought I should mention as well that with the Fusarium work, we're also trialing a pathogenomics approach to characterize the pathogen populations, because pathogen populations are constantly evolving. And in the UK, Fusarium head blight occurs as a complex of multiple Fusarium species. And to do classical mycology, so to do isolation from lots of different sites, It's very time consuming, and when you've got 1,000 reals to phenotype, you've not necessarily also got the time to collect large populations of the pathogen. So what we're doing in this work, we basically ask all our agronomists around the country, so we have about 40 sites covered, we send them tubes of RNA later, which is a solution in a tube, and ask them to put an infected grain or piece of the spike in the RNA later. We extract the RNA, run RNA-seq on that, and then we're using that to try and dissect both the pathogen and the host that it was infecting. So it's kind of a diagnostic tool. What was infecting the plant, and what was the variety that it was infecting. So we're using this as a tool to study the pathogen dynamics and try and combine that with our understanding of resistance mechanisms in the wheat crop as well. And as well as disease traits, we work on more classical agronomic traits, yield components. So this is another student who's working on yield components and ear length. And we also use some phenomics technology, some more high throughput technology to avoid Funmi having to dissect every single ear that she collects from 1,000 reals in the field. So this is a method that we're trialing, again at the National Plant Phenomics Center in IBIS. And this is based on CT imaging of intact ears. So we're able to pass the intact ear through the CT scanner and reconstruct the ear with all the grain sites in their relative positions. And from each of these grain sites, we're able to extract various metric data. So rather than threshing an ear and looking at the size ranges and distributions, we're able to do that in an intact ear, which tells us more about where the yield components or increases or decreases in yield components are actually occurring in the intact ear. And a big part of us being able to do high precision mapping in our MAGIC population was generating a genetic map. So in wheat, we're quite under-resourced, so we don't have a complete genome sequence, which hampers mapping generally. But one of the reasons for making a MAGIC population is the high levels of recombination, which allow you to map very efficiently. So our MAGIC map is now published and is the work of one of the postdocs in the group, Keith Gardner, and a PhD student, Lucas Witten. And so this was a labor of love to create our map, and I won't go into it in too much detail, but basically you have 20,000 SNP markers from a SNP array, which are variable in MAGIC, and these were used for constructing the map, and some comparisons to other maps. I guess in wheat, in all other crops, there's always a competition between whose map's better. So this table just shows you that ours is definitely the best. So we've mapped quite a large number, about 20,000 markers. And if we look at these in comparison to the previous wheat consensus map from 2014, markers in common we've actually mapped more, and this is because of the increased recombination, allowing us to map more markers to discrete positions. So here we have 4,500 sites versus 2,800 in the previous consensus map. So this is an advance over previously available genetic maps available in wheat. We also, as with everything, make it completely publicly available. So it's a new resource for the community to use. And our map is very clean. So chromosome 3b of wheat has a complete physical map. So we're able to compare it to the 3b physical map. And you can see this is our MAGIC map. So wheat, like many species, has a large, non-recombining region around the centromere. So we still see that. But when we look in comparison to the previous consensus map, we've truncated this non-recombining region via the multiple rounds of recombination. And the red dots represent errors, so we've got many fewer errors. So we've got higher precision for mapping using this highly recombined approach to generate the genetic map. And I previously showed our example of mapping this presence or absence of Orn's locus. And when we did that previously using the previous map, I think the cutoff was about 6. So now when we map this, we've got a cutoff of about a peak of about 3,000. So we're able to map these traits with very high precision in this population. And the population also gives us new opportunities. The map gives us new opportunities to understand more about the genetic structure because we impose no selection throughout the creation of the population, apart from the deselection of double dwarfs, so lines with multiple dwarfing genes, which are very short and were phenotypically eliminated from the population. So if we look, this is the signature of selection. So if we've deselected these two circled in blue, they don't correspond with high marker density. So they're distorted, but they're not corresponding with high levels of marker density. And then we see other examples where we have segregation distortion observed in addition to the presence of high levels of marker density. And these we hypothesize are the signatures of interspecific introgressions into the wheat genome. So we know there is an introgression on chromosome 1b, so breeders used crosses with rye, introducing the 1R chromosome, part of the 1R chromosome of rye into wheat, and this was very favorable for breeding and conferred a number of advantages to wheat. So we know this is the introgression from rye, so we've got segregated distorted locus and a corresponding high marker density. So we hypothesize that these other segments, which are represented in red, are also interspecific introgressions, but ones that we know little about. So this is relevant because UK breeders, breeders in Europe have commonly used wide crossing, wide hybridizations, and one great example of this is Robigus, which is a parent we use a lot, and it's a parent that still features in many of the varieties that are produced and perform very well in the UK. And so it carries, so Robigus derivatives have resistance to quite a number of diseases, some of which have broken down and some of which are still effective. And it's been thought or suggested or known kind of within breeding circles that the pedigree contained Triticum dicocoides, so this wild Emma tetraploid form of wheat, but it's never been understood or evaluated, despite many people having tried where these introgressed fragments from Robigus occur in the genome. So at this point, our work in the MAGIC population coincided with our work within the WISP program to develop this new 820K array, which also included in the validation array a number of elite varieties with Robigus in their pedigree, as well as a number of lines with Robigus not in their pedigree. So we were able to use this data set because it's publicly available to validate some of our introgression fragments from the MAGIC map. And when we do this for our biggest segregated distortion linkage block, which is on chromosome 4AL, we find that the 820K Robigus markers are a perfect fit for this fragment. So this really matches all of our predictions for an interspecific introgression. So we believe we've found the presence of this introgression, which has been known about or suggested for quite a long period of time, but not been identified to a precise genetic location. So we can then generate Cas markers very rapidly and use those to screen additional material, and then that information can be used to isolate the introgression fragments and to understand more about the effect of those in isolation. We also do more traditional biparental genetic mapping. So this is an example of a population which is also linked to Robigus. So these parents of the population, Oakley combines the Robigus introgression fragment with Gatsby, which was a terrible variety that had a different introgression from Triticum dicocoides. So this is an interesting segregating population. And we use this to study various traits. So this is an Ndvi, which we measure each week in the field, as well as we're not investing in the use of UAVs, but we use service providers who crash their own helicopters and things into trees, so we avoid doing it, and using them to assess the population and to use these traits in QTL mapping. So I'll finally talk about our work on quantitative genetics, so genetic mapping and genomic selection, which we've been focused on for the past few years. So this is an association mapping panel that we worked on, which is a European winter wheat panel of 376 varieties from France, Germany, and the UK. And it was from an EU project called the Tritici Genome Project, which was led by Catherine Foyer. and the material was phenotyped extensively. So over 2 years it was phenotyped in the UK, in Germany, and in France, and a large number of traits recorded in these experiments. And the purpose of this was to do association mapping, so to understand more about the genetic control of complex traits. At the time, we used DART markers, which for 6 sites of phenotyping data told us that PPD1 has an effect on flowering time, so perhaps we didn't need to do this experiment to show that because we already knew it. So it showed some of the limitations of genome-wide association mapping when you're limited by your number and density of genetic markers. However, we're now just finalizing the GBS data set, which gives us 10,000 markers, and we see now we have additional marker trait associations for things like flowering time. So this is on 7A is an earliness per se QTL that we've been chasing for quite a long time, and now we see that within our association mapping panel. So sometimes enriching our marker density has been very beneficial. We also use this population as an experimental population for genomic prediction. So this is work I'm doing in collaboration with a statistical geneticist at the University of Oxford. And so we basically want to use genomic selection as a mechanism to represent what a breeder would be selecting on. So if we do genomic selection just on yield, we're actually ignoring the fact that yield is the summation of many additional factors, so the flowering time, the amount of the crop that's killed in the winter, the lodging, the height, and so all of these factors are combined to contribute to yield. And equally, all of the genetic effects are influencing one another or interacting in different ways and in different directions and with different strengths. So we have stronger effects compared to weaker effects. In addition to that, we're looking at cross-site, cross-environment. So we've got different effects in the UK, in France, in Germany. So in France, we have a lot of photoperiod insensitive material. So this is affecting the maturity, the grain protein, and yield. So we're trying to build this model for multivariate genomic prediction, which incorporates multiple environments, genotype by environment effects, marker by environment effects, and is the summation of all of the traits simultaneously. And so this is very much work in progress, but at this point, you could be more, we hypothesize that you could be more confident of your genetic breeding value because it is incorporating many of these things. And many of our breeders have breeding programs or testing locations in France, Germany, and the UK. So this would enable them to combine that material in their prediction equations. And we have a new big genomic prediction project, which is actually a collaboration with all of the commercial breeders, as well as John Hickey and Gregor Gajanich at Roslin in Edinburgh. And in this, we're taking elite advanced populations from breeders, advanced breeding nurseries, and each breeder is contributing 11 of their elite crosses. And so rather than getting a prediction equation on their 11 crosses, we combine their 11 crosses, 100 individuals from each cross. So instead of having a prediction based on these 1,000 individuals, we combine the material from the 4 commercial breeders and give them back. So they put 10 in and they get the information on 40 populations back. So it's a way that we propose. We are the kind of central part of this, and they don't share material, but the prediction that they get back from the training population from the genomic prediction is based on a larger amount of material. So we're kind of the central point of that, and they put information in and get huge added value from that information. And we're also trying to do various other things, look at the interaction of environmental covariates in this process. And if you're interested in quantitative genetics, as part of some of our new projects with India, we'll be running quantitative genetics training courses in India, which will be free to attend. So I would encourage people, if they're interested in that area, to come along as well. And this is the work of the past 9 years. So this is our team. We're a relatively small team, working very hard in the glasshouse and the field to generate this material and these results. And it's really the starting point for us using our germplasm resources and our genomics capability to understand more about wheat and the genetic control of complex traits and some key collaborators in our program. That's all. Thank you.
Alison, thank you for a very nice presentation. I have a question on those transcription recombinants that you had with the yellow rust. Did you look at them more carefully? Are there any other associated phenotypes?
Yeah, so that was the first step because that was based on it was based on a seedling screen and an APR screen, but we've now, because the races of yellow rust are changing so much, we screen them for all the seedling resistances and send them to Australia for them to screen against all their different races in quarantine. So yeah, so that's where we are at the moment. So that's what's holding the publication, is to make sure that those are actually resistant progeny from the population.
I'll have a couple more questions. So, Alison, again, just a clarification. Did you mention that RHT8 dwarfism went together always with the PPD1?
Yeah, so they're linked, but it's about 20 centimorgans between them, but it's not broken in any elite material. So it has been recently reported that you can break the linkage. So Simon Griffith's group at John Innes Centre showed that they could break the linkage between them. But in practical terms, in breeding, if you use PPD-D1, you'll have reduced height from the RHT8. So they're typically, they would, in a breeding population, they would occur as a linkage.
The other one is a general question. So NIAB produces the fundamental research that goes into the UK's breeding. Right? Does research money get pumped back into NIAB?
That's a very good question. So I think we would like to see that more. And we've talked about, it's very difficult for us to capture financial return from some of the material, especially, so we get money up front from the funders to produce this public good plant breeding. You know, they like to write that it's public good plant breeding. And then we invest 7 to 8 years to develop material, hand it to the private sector, and then they market varieties. And we have no value capture mechanism from those varieties. We tried to enter into an agreement where we would have a 5-way royalty share in one of these programs. But I was saying earlier, cynically, the message back at the end of that from the breeders was it was still one cross away from a variety. So we don't capture any value from return from the breeders, apart from the fact that it is public good plant breeding, and that they'll always support the research going forward, but not financially. Although the margins in wheat breeding are very small, so it's not a kind of global. It's an inbred crop with no hybrids yet. So maybe when we have a new hybrid system, we'll be making some more money.
So in that CT scan you showed for the ears for yield components prediction. So obviously you can get the seed number and seed size. How would you get the seed weight? So do you do that by manually?
You can do the volume. So then you can do the volume of a seed, but then you have to weigh it and interpolate from the weight data. So we still do dissect the ears as they are and use that as kind of ground truth data to match the CT scan. But yeah, so if you're using volume as a proxy for weight, it's not perfect. And this is still the kind of prototype of that system. But yeah, I'd be interested to. Yeah, so we committed to making 50 of these in the proposal, and we thought it would take us a year to make 50, because CIMMYT made 1,000 in kind of a couple of years. But it's so far, we're like 6 years in and we've just made 50. So we haven't done it on a targeted basis yet, but now we have quite a lot of candidates. So we have novel vernalization alleles. So we're making more specific combinations of synthetics. But it's not a simple process, so it's not always, there's a lot of sterility and incompatibility issues. So we're still trying to overcome that to make targeted synthetics.
You mentioned about the yellow rust of wheat, and you have that segregants, transcription segregants. So do they have distance across races, or you have the specific race distance?
Yeah, so that we did that testing. So in 2011, there was a new evolved race of yellow rust in Europe, this warrior race, which obliterated most of the varietal resistance. And we did that screening with the warrior race. So it was the most common race at the moment. There's a new race as of last year. So that's, yeah, we're in the process of doing that. So we're doing all races that we have in collections at the moment. And yeah, and that's part of the validation. Yeah, so that's going on at the moment. But obviously, if we can characterize the resistances and have the markers, it's still useful even if that material doesn't yield. So we can move that into, but then obviously it's still a number of crosses away from something that a breeder can use. But I think there's enough evidence to support that if they are beneficial yellow rust resistances, that they will be taken up by the breeders into advanced breeding material. So when we started the magic population, we chose really elite parents for the population, but that was in 2007. So even now, if we identify something, it's still going to need to be moved into advanced breeding material. Yeah, so we started phenotyping at the F6 generation, but we did our genotyping F4 to F5 generation. And the advantage of doing the genotyping then is there's still heterozygosity. So when you phenotype something and you find an interesting variant, you can go back to the heterozygosity and extract these instant magic nils from that population. So that's on the phenotyping side. And the mapping approaches, I think that's still up for debate. So we use MP-Map, which is a mapping software developed by Emma Huang and the group at CSIRO. But we're also working on the Erie rice magic, diverse magic population. And there you have a slightly different situation because you have an interesting crossing scheme, which is not as structured as ours. So in ours, everything's derived from these funnels within the crossing scheme. So it's very easy to attribute that variance to the pedigree structure of the population. We're trying to work out the Erie pedigrees at the moment, but there's a lot of non-standard pedigree usage within it. So then it's very difficult to account for that population structure in the analysis. But one of the things we're doing in that population is simulating the best methods or the most powerful methods for doing the trait dissection within it. So it will be some form of QTL mapping to take into account the pedigree structure.